Good morning.
Hello, Professor John.
Hi.
Good morning to you. For us, it is good afternoon.
Right. Good afternoon.
Any of your other colleague also will join? Professor Jones?
Beg your pardon?
Any of your other colleague will also join?
I don't think so, apart from, I did invite my former postdoc, now project leader, I think in Bangalore, Ami Redkar, who's to join. So maybe he and some of his colleagues will join from his lab.
Yeah. Again, time problem is there, time zone problem.
There's Ami. I can see him on his name on the screen at least.
Yeah.
There he is. Hi, Ami. Did I get your affiliation right? Where did you move to? Remind me.
I'm based at the University of Pune.
Pune, all right.
Dr. Mandal, shall we start?
Yeah.
Many people have joined.
Dr. Kaushik?
Shall you start?
Yes.
So a very good afternoon to everyone, and of course a very good morning to Professor Jones. So we our respected director of National Rice Research Institute, Dr. Padmini Swami. Today is our distinguished speaker, Professor Jonathan D.G. Jones, Fellow of Royal Society of London, an eminent scientist and group leader at the Sainsbury Laboratory, Norwich, UK. All our heads of divisions and regional stations, scientists, students, and dignitaries from ICR and other institutes. A very warm welcome to all of you, and a very good morning to Professor Jones. We are extremely pleased to have you with us, and we are eagerly waiting for your talk today. So as you know that this special talk is a part of our lecture series being organized at National Rice Research Institute for celebrating the 75 years of independence of this country. So we call it as Azadi Ka Amrit Mahotsav, which is an initiative of the Government of India to celebrate and commemorate 75 years of progressive India, the glorious history of its people, its culture, and its achievement. The official journey of Azadi Ka Amrit Mahotsav commenced on 12th March 2021, which started with a 75-week countdown to our 75th anniversary of independence, which will end post a year on 15th August of 2022. The Mahotsav is dedicated to the people of India and will be celebrated as a people's festival in the spirit of people's participation. So the Indian Council of Agricultural Research is undertaking various activities in this regard, and these includes awareness campaigns and lecture series by eminent personalities on the thematic areas for reaching out to the maximum stakeholders in different sectors of agriculture. So under ICAR's umbrella, National Rice Research Institute is also organizing a lecture series. Today we are fortunate to have with us Professor Jonathan Jones, fellow of Royal Society of London, an eminent scientist and group leader at Sainsbury Laboratory, Norwich, UK, to deliver a special talk on dissecting and deploying plant immune receptor mechanisms for crop protection. Professor Jones is a very well-known scientist in the field of plant biology, and he needs actually he doesn't need any introduction. He has made tremendous contributions in understanding the mechanisms underlying resistance to plant disease. Professor Jones was among the first to isolate and characterize disease resistance genes. His pioneering work on plant immune receptors are being used in understanding the critical processes in plants' defense response and holds a great promise for increasing yield, decreasing the use of agrochemicals, and reducing the environmental impact of agriculture. So to begin our today's program, first, I would like to request Dr. P.C. Rath, our head of Protection Division, to welcome Professor Jones and give a brief about the program. Dr. Rath, please.
Thank you, Dr. Kaushik. Good morning, Professor Jonathan D.G. Jones and colleagues in the Sainsbury Laboratory, Norwich, United Kingdom. Good afternoon, everybody in India. I take this opportunity to extend a warm welcome to Honorable Professor Jonathan D.J. Jones of the famous Sainsbury Laboratory, Norwich, United Kingdom, to our institute, although in virtual mode, on this occasion of a special talk series to celebrate 75 years of India's independence. I welcome our Honorable Director, Dr. Padmin Swami, to preside over this virtual function. Hearty welcome to all the dignitaries of ICAR headquarters and other ICAR institutes, head of divisions, scientists, all staffs of ICAR NRI Qatar, and welcome to one and all to this virtual function. Madam, this is the 7th lecture in the series of special talks to celebrate 75th year of independence, that is the Azadi Ka Amrit Mahotsav. The first talk was delivered by Dr. P. Chandrasekhar, Director General, Manage Hyderabad, on agripreneurship-led extension on the 18th August 2021. The second talk was delivered by Professor Robert Henry of the University of Queensland, Australia, on genomics of wild and domesticated rice on 7th September 2021. The third talk was delivered by Professor Yenong Yang of Pennsylvania State University, USA, delivered on CRISPR-Cas enabled crop precision breeding and disease diagnostics on 7th December 2021. The fourth talk in this series was delivered by Professor Detlef Wingel of Max Planck Institute of Biology, Germany, delivered on the topic, a mutation is a mutation is a mutation, on 28th January 2022. The fifth lecture delivered by Dr. O.N. Singh, Vice Chancellor, Birsa Agricultural University, on topic intellectual property rights, retrospective and prospective, on 15th January 2022. The sixth lecture in this series was delivered by Dr. Carol T. Bull, Department Head, Plant Pathology and Environmental Microbiology, and Director, Penn State Microbiome Center, Pennsylvania State University, USA, on topic translational taxonomy from microbes to microbiomes on 8th March 2022. And this is the 7th talk Professor Jonathan D.J. Jones will deliver on topic dissecting and deploying plant immunoreceptor mechanism for crop protection on virtual mode. let us listen to this talk by Professor John. I welcome you all again to enjoy this special talk by Professor John. Thank you.
Well, thank you very much for all those kind remarks from
Sir,
those who introduced me.
Sir, excuse me.
Can you hear me?
Professor Jones.
Yeah.
Just a minute. Few more formalities are there. Then we'll request you to start your lecture, please. Just please wait for 5 minutes. Okay.
Okay.
Sorry, Professor Jones, to interrupt you. Just a minute. I will just.
Oh, beg your pardon. I thought I'd got to the end.
So before that, please pardon us for 5 more minutes. Yeah. So thank you, Dr. Rath. So
that's right, because we're not on schedule.
Okay.
We're ahead of schedule. Well, I can still say thank you very much indeed for those very kind introductory remarks.
Okay.
And it's good that you heard from Detlef Weigel, because I imagine he would have also talked about plant immune receptors, but in a slightly different context, but also probably emphasizing plant immune receptor genetic diversity. Am I right in that? Maybe not. Yeah. Anyway, so
okay, we are quickly coming back to you, Professor Jones. Before we start our talk, I would just request our director, Dr. Padmini Madam, to have a very brief opening remark. And after that, we will have the speaker will be introduced by Dr. Sudhamay Mandal, and then we will quickly go towards Professor Jones for the lecture we are waiting for. Madam, please.
Sorry if I ruined your program.
Yeah. No problem. No problem.
Okay, that's it. Okay, fine.
Madam, please unmute yourself.
So first, I beg excuse, Professor Jones, because I interrupted in between as I have a very important meeting in our council after half an hour. So I will leave for some time. So my thing they brought to front. So I beg excuse. A very warm welcome to Professor Jonathan D.G. Jones, Director, Indian Institute of Rice Research, Hyderabad, Dr. R.M. Sundaram, all dignitaries joined in the meeting, all head of the divisions, all scientist colleagues from NRRI, IIRR, and other ICAR institutes, and also other organizations, research scholars, students, and ladies and gentlemen. I know you all are eagerly waiting for this special talk will be delivered by Professor Jonathan D.G. Jones, Dissecting and Deploying Plant Immune Receptor Mechanism for Crop Protection. A year ago, in March 2021, a news in TSL website captioned, "Plant immunity requires two-step detection of invaders," referring to a paper by Professor Jonathan Jones' group. This news sums up that cell surface and intracellular receptor of plant work together to activate a strong defense against bacterial pathogens. Professor Jones has an illustrious scientific career that may inspire a whole generation of scientists. Indeed, probably he is the most highly cited plant scientist in the world with more than 140,000 citations. The Jonathan Jones group is interested in how plants resist disease and how resistance is overcome by pathogens. To suppress host defense, pathogens secrete effector proteins into plant cell or into their apoplastic spaces, how these effectors work, and identity of their targets in the host help to understand plant defense mechanism, etc. Since 1988, Professor Jones has worked at the Sainsbury Laboratory, very famous laboratory in Norwich, UK, serving as head of laboratory 1994 to 97, again 2003 to 2009. The TSL is an independent research institute that focuses on plant health for a sustainable future. It makes fundamental scientific discoveries in molecular plant microbe interactions and applies these to reduce crop losses caused by plant diseases, particularly in low-income countries. Established in 1987, highlights of TSL include first discovery of RNA interference in plants by Professor Sir David Balcom, FRS, and second discovery of the first immune receptor in plants by Professor Jonathan Jones, FRS, as recognized by the University of Minnesota Stockman Prize. Through the development of novel approaches in genetics and immune receptor discovery, Professor Jones and his colleagues, they developed late blight resistant potato cultivars, tested those in UK fields, and developed commercial crops through partnership with Simplot, a US-based potato breeding company. These new potato lines of Maris Piper could potentially save millions of pounds. Understanding the plant immune system is crucial for using genetics to protect crops from diseases. Plant resist pathogens via two-tiered innate immune detection and response system. The first plant resistant gene was cloned in 1992. Since then, many cell surface pattern recognition receptors have been identified, and the genes, resistant genes that encode intracellular nucleotide binding leucine-rich repeat receptors, NLRS, have been cloned. In addition to immune receptor, many components of immune signaling networks were discovered over the last 30 years. Recent studies have reinforced the importance of interaction between the two immune systems. Professor Jones will provide an overview of interactions between PRR and NLR mediated immunity, highlighting challenges and perspectives for future research. I wish his lecture will enrich our scientists on plant immune receptor biology that holds so high application potential for the entire humankind. Again, I congratulate Professor Jones and welcome him for this auspicious function. Particularly, the country is celebrating Azadi Ka Amrit Mahotsav, and we, along with the country, whole country, in the staff of ICAR, we are all also joining hands to celebrate this. And as a research organization, we are organizing, as Dr. Pisira told, so many lecture series in scientific research. Not only we, all the ICAR institutes, and this is also a auspicious day for us to have Professor Jones with us. And I again welcome him for his very eloquent lecture, which he is going to be excited to deliver, that is dissecting and deploying plant immune receptor mechanism for crop protection. I again welcome you all to this gathering, and I hope all will enjoy his lecture. Thank you, Dr. Kaushik, and Dr. Mandal, carry over.
Thank you, madam. Now I will request Dr. Sudhama Mandal, Principal Scientist and Convener of this program, to quickly introduce our today's distinguished speaker to the august audience who are present virtually.
Thank you, Dr. Kaushik Chakraborty. I will take just a couple of minutes to introduce Professor Jonathan D.G. Jones. It's my pleasure to introduce our special guest to the August virtual gathering, Jonathan Dallas George Jones, popularly known as JJ. The name itself speaks volumes in the scientific community, commanding about 140,000 citations in scientific literature. Professor Jones is a plant molecular geneticist who has made distinctive contributions to understanding how plants resist disease and to how pathogens circumvent host immune mechanisms. He was born in London in 1954, where he grew up. He graduated from Cambridge University with a degree in botany 1976 and a PhD jointly between Cambridge Genetics Department and the Plant Breeding Institute in Trumpington 1980. After postdoctoral work with legendary Fred Ausubel at Harvard on symbiotic nitrogen fixation 1981 to 82, He worked at a startup AG biotech company, AGS, in Auckland, USA, working closely with Hugo Dunner to study the behavior of maize transpositions in tobacco. Since 1988, JJ has worked at the famous Sainsbury Laboratory, TSL, Norwich, UK, serving as the head of laboratory during 1994-97 and again 2003 to 2009. He was elected member of the European Molecular Biology Organization, EMBO, in 1998, Fellow of Royal Society in 2003, and international member of the US National Academy of Sciences in 2015. In 2012, he was awarded the prestigious Minnesota Stackman Prize. He is the professor at the University of East Anglia. He has served as advisor to the Danforth Center in St. Louis, USA, and is an advisor to the Two Blades Foundation and to King Abdullah University of Science and Technology in Saudi Arabia. I'm tempted to mention here that at AGS in Auckland, Professor Jones worked alongside plant scientist Caroline Dean, now his wife. Dean, Dame Commander of the British Empire and an elected foreign associate of National Academy of Sciences, USA, has advanced understanding of vernalization and the molecular control of flowering. Professor Jones was among the first to isolate and characterize disease-resistant genes. His discovery of receptor-like proteins, RLPs, preceded discovery of leucine-rich repeat innate immune receptors in animals. Jones' work also revealed intracellular nucleotide binding leucine-rich repeat NLR immune receptors, which often function in pairs to detect pathogen molecules known as effector proteins to activate defense. With biologist Jeffrey Dangle, Jones authored two highly cited review papers published in Nature in 2001 and 2006, respectively. The first review paper outlined the GARD hypothesis, while the second highlighted integrated thinking about cell surface triggered immunity and intracellular receptor triggered effector triggered immunity, or ETI, into one unified model, the famous zigzag-zig model. His current research interest includes paired NLR immune receptor function. The Arabidopsis RPS4 and RRS1 genes encode an intracellular immune receptor complex that is required for recognition of bacterial effectors, AVR RPS4 and POP-P2 via their interactions with RRS1, WRKY DNA binding integrated decoy domain. The Jones lab is investigating how recognition of effectors results in defense activation by the complex. And the second aspect he researches on is novel resistance genes to P infestans. The Umycete Phytophthora infestans, as we know, causes potato late blight that once resulted in the infamous Irish famine in 1840s. The Jones lab is isolating novel resistance to P infestans, RPI genes from wild diploid potatoes, notably Solanum americanum, shorts of RPI AMR1 and RPI AMR3. RPI VNT1 is the first such resistance gene deployed transgenically in commerce. To accelerate R gene cloning, the Jones lab developed RENSEQ, R gene enrichment sequencing, a method to define all the intracellular immune receptor sequences, the NLROM, of a plant of interest to reveal receptor diversity and evolution. The Jones lab studies how RPI gene encoded NLR immune receptors detect pathogen molecules and activate defense, and also whether deployment of such genes in gene stacks provides durable crop disease resistance. I come to an end with Professor Jones' words. We aim to reduce crop losses to disease using genetics. This involves public engagement, making the case for rational exploitation of genetic diversity to reduce the environmental impact of agriculture, and also, of course, defending the use of GM method and of gene editing to bring this about. Thank you. Over to Dr. Kaushik Chakraborty now.
Kaushik.
Thank you, sir.
Dr. Kaushik.
Yes, madam.
We have Dr. Sundaram also with us, Director IIRR. So
Yeah.
I think I will just request him for a few lines. Dr. Sundaram, are you here? Dr. Sundaram.
Dr. Sundaram's on mute.
Yeah.
I think we can also take Dr. Sundaram's remark at the end of the lecture if he is not available currently. So thank you, Dr. Mandal, for introducing our speaker. And now we are. I invite Professor Jones for the special talk for which we are eagerly waiting. And also I apologize to him for the little bit change in our proceedings for which the confusion created. So Professor Jones, please.
Well, thanks again for all those very kind remarks about me, which I'm very embarrassed by, really. Very flattering. Hardly recognize this person that you're introducing. I'd also like to say for congratulations to India, 75 years free of the British. Well done. But of course, when it comes to food security, you're on your own. So I hope you'll plot your own path in terms of regulatory policy and think about what's really the best interest of India and not be governed by the delicate sensibilities of people in Europe. Right. So let me click share screen. Okay, can everybody see that okay?
Yes, sir.
Yeah, it's
and
fine.
You can hear me okay. Well, you've got a sneak preview of much of what I'm going to say from these various kind introductory remarks, but I'll be building out the argument in a bit more detail than you've heard. So what we want to do, as you've heard, is we want to replace chemistry for crop protection with genetics, and we certainly want to achieve better crop protection. It's often said, for example, that you need 1,000 tons of water to produce a ton of wheat. So if you have a 10% yield loss to disease, that's 100 tons of water you've wasted. It's still been applied, but it hasn't led to production. So you really want to protect crops from disease. And in order to protect crops from disease, you have to understand the mechanisms at play in the interactions between plants and their pathogens. So it's often said that most plants are resistant to most plant pathogens. So most plants are actually poor hosts to most of the microbes that have evolved to colonize plants. And why is that? Plants have actually got really good defense mechanisms. They activate production of reactive oxygen species and synthesis of antimicrobial peptides. They activate cell wall thickening, fortification. They activate the production of secondary metabolites, specialized molecules that act as antibiotics against microbes. So when you have disease, how does the pathogen achieve that? Well, it achieves it by evading detection. So basically, if a plant knows that a pathogen has showed up, it can activate defense pretty effectively. If I can make this, I don't know if you can see this. But in order to activate those defenses, it has to detect the pathogen, either through cell surface receptors or intracellular receptors. So the surface receptors detect molecules that are relatively conserved from the pathogen. I hope you can see my little arrow wiggling around here. By cell surface receptors here, detecting apoplastic molecules. Also, there are intracellular receptors that detect molecules that the pathogen has made, they're delivered either by bacterial type 3 secretion or specialized structure called the haustoria to deliver effectors from fungi and oomycetes. These effector molecules have evolved usually, but not exclusively, to interfere with host surface receptor initiated defense mechanisms. Some of them also, like TAL effectors, promote pathogen nutrition. But if the plant has evolved the capacity to detect an effector, then you get what we call effector triggered immunity, or intracellular receptor mediated immunity. So basically, plants have got good defense mechanisms, but they need to be activated by one or both of these two classes of receptors. Right. So what about the intracellular receptors? I'm not going to say much about the cell surface receptors. So there's a class of molecules out there in nature that have a sort of, let's say, primarily can be divided into 3 domains. And they all have a nucleotide binding domain in the middle. In plants, they usually have leucine-rich repeat domains at the C-terminus and 3 different flavors of signaling domain at the N-terminus. And most resistance genes actually encode proteins with this kind of structure. We used to call them nibblers, nucleotide binding leucine-rich repeat proteins, but now to align our nomenclature with the mammalian field, we call them NLRs, nucleotide binding leucine-rich repeat immune receptors. And it turns out that animals and fungi have similar structured molecules. There's a bit more variation in the kind of nucleotide binding domain, the kind of domains at the N-terminus, kind of domains towards the C-terminus in animals, and even more diversity in fungi. For many of these, and especially in fungi, it's not clear what the function is. Some are involved in strain-specific mating compatibility. But in plants, there are the 3 main types. They've got either got a coiled coil at the N-terminus, or what's called a tear domain, total interleukin-1 receptor domain at the N-terminus, or there's some that have an RPW8 domain, which is actually a specialized kind of coiled coil domain. Now, the first structure determined for a protein of this type was actually a mammalian protein involved in cell death called APAF1. It makes what's called an apoptosome because it promotes apoptosis. And it's a pentamer of these kind of molecules. And what it does is by oligomerizing, it imposes induced proximity on the N-terminal domain, which in this case is a CARD, the caspase recruitment domain that recruits caspases. And what happens when they're all brought together is you get 2 caspases or more next door to each other, and they cross-proteolyze each other, and then you get irreversible activation of signaling. The same goes through for this worm protein, C. elegans protein. Then there's a mammalian so-called inflammasome called NLRC4, one of the first to be characterized, a very beautiful structure involving 1 copy of one protein and 10 copies of another. And again, it imposes induced proximity on a caspase recruitment domain. And about, I think it might be 3 years ago, at least 2 years ago, the first plant NLR protein oligomer was defined, a so-called resistosome, a Arabidopsis protein called ZAR1. And what it does is to oligomerize and impose induced proximity on the C-terminal coiled coil domain. And what this does actually is to create a cation channel that activates signaling. And this is recognition dependent. It's independent, it's indirect recognition, but anyway, the outcome is oligomerization. Subsequently, it was shown for a couple of different tier NLRs, ROC1 and RPP1. ROC1 is a Nicotiana benthamyana protein that recognizes a bacterial effector, ZOPQ. RPP1 is an Arabidopsis protein that recognizes an oomycete effector called ATR1. And when they are convert from the inactive to the active state, what you get is a tetramer, which imposes induced proximity on 4 copies of the tier domain. And this enables it to activate an enzymatic activity that's intrinsic to an oligomer of the tier domain, but absent from the monomer. And that enzymatic activity is an NADase that almost certainly releases molecules that activate downstream signaling. So again, the principle applies that basically what these NLR proteins are is little detectors which, when they engage with a recognized molecule, so here ZOPQ, for example, is a recognized molecule, engages with the leucine-rich repeat and also a domain beyond the leucine-rich repeat. Conformational change, the nucleotide binding domain ceases to bind ADP, but starts to bind ATP, and that imposes a conformational change on that nucleotide binding domain that gives you the oligomer. It means it can now associate with other monomers of the protomers of that protein, and you get the tetramer, and you get the enzymatic activity, and you get downstream signaling. And again, it's irreversible. So induced tear proximity activates the NADase activity. So we'd actually know an enormous amount now compared to certainly when I started 33 years ago, or even compared to 5 years ago, on how these NLR proteins work physiologically to activate downstream signaling. So to sum up, I don't know if this is getting in the way or not, but the plant and animal NLRs bind ATP upon activation and oligomerize, imposing induced proximity on an internal signaling domain. And tear domains have NADase activity. The oligomerized coil domains often create cation channels that can let calcium in to activate signaling. What I haven't said anything about is that most plant sensor NLRs actually require a so-called helper NLR to activate defense. So they don't entirely activate defense on their own. All the tier NLRs require, for example, the RPW8, that third class of NLR, the defense activation. I don't want to get into too much detail about that in this talk. So that tells you a bit about how the proteins work, but how do they work in natural populations? And within natural populations of plants, before domestication, before we created genetic monocultures, our genes actually worked via frequency-dependent selection, I would argue. You don't get monocultures of one resistance gene set in natural populations. You get a lot of diversity. And that means that a pathogen that has evolved to evade one resistance gene on one plant in the population most probably has not evolved to evade other resistance genes in other plants in the population. So that very diversity greatly restricts pathogen success. So NLRs that give race-specific resistance, I would argue, operate usually under frequency-dependent selection in the wild ancestors. And that gives weak selection for the pathogen to evolve to evade that recognition, because even if they do evolve to evade it, they'll have lost an effector, and it won't, having done so, will not help them when they try and grow on many of the other plants in that population. And the outcome of this is you've got great selection for NLR allelic diversity, and you also have some selection in the pathogen for diversity in the repertoire of effectors. So although often people talk about an arms race between plants and pathogens, I would argue that actually in nature, it's not an arms race. It's more like trench warfare. If you're a plant breeder, it feels like an arms race because you are required to produce varieties that meet criteria of distinctness, uniformity, and stability. But that puts you in a difficult position, because if you put out 1 million plants that are genetically identical, then any pathogen that can get in there does very well. And I often depict it with this visual aid. So just imagine you've got an R gene monoculture. So let's say one color represents one recognition capacity. Of course, in most plants, they've got hundreds of NLR genes. Rice has got 400 to 600. But anyway, so for simplicity, I've depicted one R gene type here. So here's your monoculture. Every plant's got the same resistance gene. Over here is more of a natural situation. You've got diversity of recognition capacity represented by the diversity of colors. So what you know is if you've got a pathogen that can succeed on one of them, then it can succeed on all of them. And unless you apply the right chemistry, your field is toast. Whereas if you've got infection on one plant in a heterogeneous population, not only can that pathogen not grow on all of these plants, but it can grow on some of them. In this depiction, 1 in 5 plants. But the epidemic is going to be slower because first of all, the inoculum is going to be lower, and then it's got further to go to find something it can grow on. So you greatly restrict losses. And I would say I could add that I'm not saying much about rice, I'm afraid, but we do have a project with Thomas Croy in Montpellier and his colleagues, and with his colleagues in the Yongming Terraces in southwest China, where we've applied the Renseek method to land races of rice. These land races show very low losses to rice blast, but any monoculture or any variety that is introduced there is hit very hard by blast. And it turns out that when we apply the Renseek method on the rice NLR immune receptors, we see within the land race very pronounced genetic diversity of NLR alleles and in presence and absence polymorphism. So those land races, although they're called a land race, actually are show a lot of heterogeneity for NLR composition. Right, so that's all I'm going to say about that. I'm going to extend this, disease resistance notion from within species to between species. So some plants are spoken of as non-hosts for particular pathogens. So why are non-host plants not good hosts? So as I've said, most plants resist most plant pathogens. So barley's powdery mildew or striped rust will not grow on wheat, at least usually. And wheat, striped rust and powdery mildew will not grow on barley. And of course, neither will grow on rice. That's non-host resistance. Can we do genetics on non-host resistance? And if you think about the evolution of pathogens of wheat and barley from the common ancestor of wheat and barley, say 20 million years ago, they will have diverged with their hosts. So it's not hard to imagine that what you need to be successful on barley and evade detection by barley is not going to be the same repertoire of effectors as what you need to grow on wheat and evade detection by wheat. So the expectation might be that barley races can't grow on wheat because they get detected, in which case non-host resistance is the flip side of specialization, or it is special because of specialization of different races on wheat or barley. But we could ask the question, since I've referred to effector-triggered immunity and pattern-triggered immunity from intracellular and surface receptors, is non-host resistance just simply due to ETI plus PTI, or both? Or could it be an entirely different mechanism? So the question is, how do you do genetics on non-host resistance? Because if everybody's resistant, then you can't do genetics. Or it's harder to do genetics. But what we'd like to do is to understand non-host resistance well enough to turn our crops into non-hosts for their pathogens. So maybe it's worth thinking about some of your important pathogens, what might be a closely related enough plant to rice that might be a source of genes for non-host resistance to these rice pathogens. At least the examples we've looked at, we think we can, because we can move perception capacities from one plant species to another. And the sort of paradigmatic paper on this, to my mind, is this work from the Staskovitz lab. You might want to include him in your list of distinguished speakers. He's got a good story to tell at the moment, in fact, several. But he cloned a pepper gene called BS2 for xanthomonas resistance. And I'm sure xanthomonas is an important disease of tomato and pepper and in humid subtropics. And this gene, you can clone out, take out a pepper, and you can confer resistance to bacterial spot disease in tomato. There's not much genetic variation in tomato for resistance to this disease, but pepper actually is quite a good source of resistance to this disease for tomato. So we chose this slightly unusual model system for our investigations of this. We investigated Albugo candida, which is an oomycete pathogen that causes white rust disease of Brassicaceae. And again, there's multiple races of this, and each is specialized on a different Brassicaceae host species. And if you want to know more about this, you could always ask Ami Redkar, who's visiting Pune at the moment and was involved in this project. So all Arabidopsis accessions resist Brassica infecting races albumin cancers such as this one, AC2V. And this is quite an important disease of Brassica juncea throughout India. And of course, Brassica juncea is quite an important oil seed in relatively hot, dry conditions. So we actually have, cloned genes that would solve the problem if only you had a fit-for-purpose regulatory framework for authorizing the deployment of crops improved using the GM method. So I mentioned this problem, right? If everybody's resistant, all Arabidopsis accessions are resistant, how do you use genetics when everybody's resistant? And the answer is transgressive segregation. What does that mean? Well, if you imagine, let's say Arabidopsis accession, Colombia has 3 resistance genes. It actually has more, but anyway, imagine it has 3 genes, A, B, and C, and another accession, let's say Nidazins, has 3 resistance genes, but they're different, called D, E, and F. If you cross those 2 accessions and self, then 1 in 4,000 will be transgressively segregating and will have lost the resistant alleles of all of those 3 loci, assuming they're unlinked. And then you'll have a susceptible. And once you've got a susceptible, then you can start doing genetics. That's basically what we did. The details of this, you can see here. Ami's a co-author. It was in PNAS, I think, about 2 years ago. So you can see the genes that contribute to resistance to Brassica infecting races of white rust and clone them. And we've had some more advances since then. So we published these 2 papers, and this one is still in the works, but we're going to resubmit the final resubmission this week, aren't we, Ami? Right. So from all that work, let's just recapitulate. We defined 4 different resistance genes that come out of Arabidopsis and confer resistance to race AC2V, and actually confer resistance in Brassica species. And so I think with that stack of 4 genes, there's a very good chance that you would render, you provide durable resistance in Brassica juncea to white rust. And white rust causes problems itself, but the big thing about white rust is it imposes very severe susceptibility to other diseases. And so your white rust infected Brassica juncea then becomes very, very susceptible to downy mildews. So you can fix that. Right. So where I am sort of, my main applied project is more on Phytophthora infestans. Phytophthora infestans can convert a field like this, the A over here, to a field like this, the C over here, in about 2 weeks. There's not, once it gets in, you see the symptoms that you see in B, there's really nothing you can do about it. And you get losses not just of the plants and the photosynthate, but also the tubers can rot too. You get tuber blight. So we've cloned genes for resistance from wild relatives. This is the first one we cloned, actually, RPI-VNT1. And you can see that in the field, I usually say at this point, here's something you don't see very often, a GM field trial in Europe. And this is a GM field trial in the John Innes Center site. And these plants are resistant to late blight, and these plants are susceptible. They're both variety Desiree. This one's had an added gene, RPI-VNT1, and this one hasn't. But of course, one resistance gene is going to be easy for the pathogen to overcome. It's not enough, so you need more resistance genes. So we go to a plant called Solanum americanum. We sometimes refer to this as the Making Americanum Great Again project. So Solanum americanum, it turns out, is an excellent source of new RPI genes for resistance to P. infestans. Nearly all accessions are resistant in detached leaf assays, and all are resistant in the field, even the ones that are susceptible if we make them provide very pathogen-favoring conditions. It's a worldwide inbreeding diploid, self-compatible, so you don't have a lot of the problems you have with some diploid solanums. It's a 1 gigabase genome, a lot of genetic diversity. It's the parent of Solanum nigrum, and we've also identified resistance to Phytophthora capsicii, Phytophthora palmivera, and nematodes. So it's a great genetic resource. Here are some growing out in the field in the plots in Norwich. So how many RPI-MR genes can we find? And can we use them to turn potato into non-host P. infestans? That's the aspiration. So most of these accessions resist P. infestans in detached leaf assays. So day 0, day 6, basically here's a susceptible, here's a resistant, and these are in very high humidity conditions, and we load a lot of P. infestans spores, so if there's any susceptibility, we'll see it. So 44 out of 54 accessions are highly resistant, 5 were susceptible, and a few showed an intermediate phenotype. And so here's a little list of most of these accessions. We cloned RPI-MR3 from this accession, and that was 2016. This is the first combination of RENseq with PacBio sequencing. And then we cloned RPI-MR1. This is reported this time last year, approximately. This is a very broad spectrum resistance. 2271 is our susceptible that we usually use. So 3 and 1 confer P. infestans resistance in benth and in potato. AMR1 is very widespread and it's surprisingly polymorphic, but they all recognize the same effector, even though all these alleles. They're all dependent on another helper NLR class called NRC, which I won't have time to talk about today, the CC-NLRs. Here's a bit more on this claim that it's broad spectrum. So there's 19 tested P. infestans isolates reported here. And RPI-MR1, 2 different alleles confer resistance to all of them, which these races cause disease on susceptible non-transgenic Maris piper. And it's unusual for an R gene to have quite such broad spectrum. The same is true of RPI-MR3. So then we can ask the question, to what extent is this non-host resistance of Americanum to P. infestans solely due to these? What other genes can we find? So part of our approach to this was to clone the recognized effectors for 1 and 3. And then we could find by transient expression into all these different accessions using agrobacterium, which recognize 1 and 3. Actually, most of them do, but some don't. And parenthetically, there's recognized homologs of AVR-AMR3 and AMR1 in many Phytophthora species. And it turns out this is quite useful because Phytophthora palmivera and also Phytophthora parasitica are again Phytophthora diseases that cause problems throughout the humid subtropics. And RPI-MR3 recognizes the AVR-AMR3 homolog from Phytophthora palmivera, and it actually confers resistance to Phytophthora palmivera as well as to infestans and to parasitica. So, clone 1 gene, get 2 free. And that work is described in a bioRxiv preprint that we're working up for resubmission to a real journal soon. So how do we find more RPI genes from Americanum? Because we know there are more there. So one thing we can do is we can look for resistant accessions that lack RPI-MR1 and RPI-MR3 and are still resistant. So these 2 accessions, 2275 and 3370, they're both resistant. They've got a locus we call RPI-MR5. They don't recognize 1 and 3, so it has to be something new, because what you certainly don't want to do is spend ages map-based cloning something you've already cloned. So we avoid that with only chasing something where 1 and 3 are not recognized. So that's one approach. Another is to look for resistant accessions with 1 and 3 that do more than 16 to 1 or 3 to 1 in resistance. So we got 3 accessions here that we're doing that for. And also I think the most interesting thing we're doing is, from a science standpoint, is looking for weak RPI genes that could contribute to non-host resistance. If you've got a stack of weak resistance genes, again, there's not much selection for the pathogen to evade them, and stacks give you more resistance. So we used effectoromics and genome-wide association mapping and to identify additional effector recognitions. That's what I'm going to tell you about now. So as a backdrop to this, we have, and these will be a publicly available resource that'll be quite useful for Solon AC students. We've got very high quality genome sequences now for these 4 accessions. So partly by collaboration with Sam Wang's lab and partly by collaboration with Ki Sohn's lab in Korea. And we know they're good quality because with the high-C analysis of the genomes, we can see very clean chromosome level assemblies. So they're good quality assemblies. And you can sort of find from characterizing all the genes and comparing them to other genes, you can see Americanum is a bit more distance from tomato and potato than they are from each other. But it's a SLAM species. It's more closely related to those two than is eggplant, brinjal. Right, and then so if we actually did a 10 deep skim sequencing with PCR-free sequencing, and so here's all the relatedness of all of our accessions. You can see that the distinct clades. There's these 6 guys are all, which are the ones that are susceptible or moderate susceptible, are all quite closely related to each other and distant from these others. So when I'm talking about effectoromics, what do I mean? So we've got all these different accessions here, and we've got about 350 RXLR effectors from the pathogen cloned into an expression vector. You can predict oomycete effectors, or at least Phytophthora effectors, if they by presence of some motifs. So we've got 350 candidate effectors. So we basically, for each effector, we infiltrate all 54, and then you do the next one, and then we do the next one. And so you can see the kind of phenotype you get if it's recognized or not recognized. And so we build a matrix like this. So these are the effectors along the top. Here's the accessions. You can see some effectors give you a response in all of them. Maybe it's not very specific, but there's a lot of mixed responsiveness. So most effectors don't trigger HR on most accessions, but a subset show interesting differential responses, and when we're investigating 8 of them. And one of them, because we had sequence data from all 54 accessions, we could look for associations between alleles and phenotype. And one of the responsiveness, 22825, is the name of the effector, the responsiveness to it appeared to map to this position on chromosome 1 by 2 different computational methods. And then we could do genetics on it. And guess what? It mapped to chromosome 1. So we could chase it down using RENSI, and it mapped to an NLR, which is actually an RPI-MR3 homolog or paralog on chromosome 1. And this is a phylogeny of the RPI-MR3 clade, and this is this guy, which does not recognize RPI-MR3. It's a homolog of RPI-MR3, but it recognizes a different effector than RPI-MR3 recognizes, this obviously recognizing AVA-MR3. So we got this weak recognition, or we got this recognition of an effector. So then when we went back to the phenotype in resistance, so this effector, when transiently expressed in Benthamiana, gives you elevated resistance. So here's the positive control, RPI-MR3, transiently expressed in Benthamiana, you get this much growth of the pathogen, i.e. not very much. For the, we call it R22825, we get this much. And if we infiltrate something that's completely non-functional, then we get this much growth of the pathogen. So you can see that just by cloning effector recognitions, we've cloned a weak resistance gene. And we've got at least 1 more already cloned. And when we've got 2 or 3 of these, we'll be making a stack and transforming to see whether the combination of multiple weak resistance genes gives you as much resistance as 1 strong resistance genes, possibly more durably. Now I talked about, and of course it was mentioned in at least one of the introductions, that we've been making gene stacks for resistance. So we've got potato lines transformed with a stack of VNT1, AMR3, and AMR1, deriving from these different hosts. And here's the stack. Basically, it's completely resistant. It barely notices Phytophthora infestans. To overcome it, you'd have to evade all three of these genes. We've got lines with no yield penalty, and they also have complete tuber blight resistance. This is a collaboration with a couple of companies. And with Simplot, we've also plugged in a tuber trait improvement RNAi module, silencing polyphenol oxidase and an invertase. And so so these lines we call Piper Plus don't brown when you cut them and when you incubate with catechol, and also have reduced levels of reducing sugars if you incubate in the cold, and that's quite a valuable trait in the potato industry. However, what we found is all of these lines we were working with were susceptible to potato virus Y. Potato virus Y is a potavirus. It has a very potent silencing suppressor that renders the plant susceptible to a lot of other viruses. So that's a problem. So we've basically, and we just got funding for this, to make lines that also have the RY gene that gives extreme resistance to PVY in a paper we published a couple of years ago. So we'll be coming up with PVY resistant versions of this. That's still, that's in the works. That's over the next couple of years. And we've also added 3 genes for resistance to a disease which I suspect is probably quite important in India, bacterial wilt, Ralstonia. Ralstonia hits eggplant, tomato, potato, and the plant yield is very severely affected. And of course, once it's in the soil, it's hard to get rid of. So this stack of 3 genes, surface receptor and 2 NLR-based immune signaling pathways, we are confident will give us great resistance to bacterial wilt. This is a collaboration with Mark Ghislaine and funded by the Royal Society. So here's the question: can we take potato off the co-evolutionary treadmill with P. infestans? You might want to ask yourselves if you could take rice off the co-evolutionary treadmill with BLAST. I think we can for P. infestans. I think we've cloned enough genes that we can make a stack that we could. So we can just get into that in discussion. So we've still got a lot more to do, isolating novel effector response genes and cloning additional genes for P. infestans resistance. But the more we clone, the stronger the stack will be. The non-host resistance to P. infestans in S. americanum is likely mostly due to the additive effect of strong and weak recognition of effectors by NLRs. Right. So what about resistance mechanistically? We tend to think genetically about resistance like it's a switch. You know, resistance is either on or off. But is it more like a dimmer switch? Is it you get more or less resistance? And of course, resistance actually is quite quantitative. So even in a susceptible plant, there's a lot of resistance being activated. It's just not enough to stop the pathogen. So people talk about basal resistance. So I would define basal resistance as like this: it's the sum of all the PTIs going on in an infection minus the sum of all the effector-triggered susceptibilities going on in an infection. And if there's any weak resistance gene activity, effector-triggered immunity is the sum of all them. So that's basal resistance. Now, it's important to remember that resistance genes are usually, at least, semi-dominant. So that's a very profoundly important thing to think about. So what it means is, you know, resistance genes are usually not very highly expressed, right? So if you halve the amount of expression in the heterozygote and you get half the amount of resistance, it tells you that the amount of resistance is gated through the amount of that receptor. If you can increase the amount of that receptor, you get more resistance. And here's the sort of classic zigzag, zigzag, zig. If you elevate the amount of pattern-triggered immunity, you might expect you get more resistance. Does that happen? Well, yes, it does, because we know this from a paper we published from work started when Cyril Zipfel was a postdoc with me, where we moved EFR, which is a surface receptor that recognizes a bacterial molecule, EFTU, a recognition capacity that's present in the Brassicaceae but absent from the Solanaceae. So we move this recognition capacity into tomato, and we convert the tomato from something that's bacterial wilt susceptible something that's bacterial wilt resistant. So all we've done is we just increases the amount of PTI by adding an additional amount of recognition. And similarly for the NLR immune receptors, so here's the transgenic potato line. This is days delay in sporulation of Phytophthora infestans. VNT1 alone, you get like a 48-day delay. CHC1 alone, you get like a 35-day delay or whatever it is. But if you've got the stack, then basically you don't see any sporulation. So more doses, stronger resistance. So we started to investigate this rather indirectly in the course of our studies on the RPS4-RS1 immune receptor, which is a combination of two proteins, one of which has an integrated domain, a Werke transcription factor domain. Werke transcription factors are implicated in plant defense. And a couple of effectors have evolved, probably many effectors have evolved actually, that have their task for the pathogen is to mess up the function of Werke transcription factor domains and resulting in virulence. So what this immune receptor does is make it the case that every time an effector shows up to hit a Werke transcription factor domain, it can't help but hit the domain that's in the immune receptor and activate the immune receptor. Okay. Yeah, and there's a lot of published literature on this, some of it from our lab with more details, but that's the concept. And there's a lot of other integrated domain containing resistance proteins with different integrated domains that are mimics for different targets of pathogen effectors. So we got into this by studying, trying to figure out what was going on with RPS4-RS1 defense activation. And we wanted to investigate we asked ourselves, how does this immune receptor do what it does? Well, what does it do? So we had to try and identify the earliest thing it did. The earliest thing it does that we can define is activate gene induction. So if you infiltrate Arabidopsis accession with RPS4-RS1 in this case, POP2, after 24 hours, you get this hypersensitive response defense activation. And a mutant POP2 or an RS1 mutant doesn't give you that. So we want to know what happened. Look earlier with expression profiling. So after 4 hours, 6 hours, and 8 hours, in the control, let's start with the control, always a good idea, you get some defense activation, actually, and that's just pattern-triggered immunity. You've infiltrated Pseudomonas that delivers this effector, but it's a mutant allele of the effector, so it's not recognized. But if you deliver a functional allele of POP2, then you get much more induction of these genes. What are these genes? ICS1 and EDS5 are involved in making salicylic acid. PR1 responds to salicylic acid, and you can see this very nicely here. PR1 shows no response early, but ICS1 and EDS5 come up. So by 6 hours and 8 hours, salicylic acid has accumulated, and then you get lots of PR1 gene induction. That's PTI plus ETI. Okay, and we call this salicylate triggered immunity, SATI. So what this tells you is that most experiments reported in the literature where people tell you they're studying ETI, they're not telling you it's studying ETI, they're studying PTI plus ETI. They're studying both. So the question then arises, how do you study ETI alone? And the answer is, we made a chemically inducible AVR-RPS4 line. So estradiol-inducible AVR-RPS4 gives you induction of ETI without induction of PTI. So if we induce AVR-RPS4, after 2 hours, we got this much peak, and then after 4 hours, we got these response genes coming up, and then after 8 hours, we got PR1 coming up. So now we're studying ETI without PTI, and we can ask, so okay, if we activate ETI alone, can we study chemically inducible hypersensitive response? Well, the answer is no, we can't, because we don't get a hypersensitive response if we only induce AVR-RPS4. So we know a lot about processes that are activated in PTI, all the various protein kinases are activated in DPH oxidase, etc. There's actually very little information, well, there was very little information on what's activated by ETI alone. And of course, there's different NLRs, so are all ETIs the same, depending, from different NLRs? And what's the relationship between ETI and PTI? And so, as was alluded to in the introduction, April last year, nearly a year ago, we published this paper, mutual potentiation paper, and basically a parallel paper with a similar punchline came out from Shufang's lab, and we coordinated the appearance of this in the literature, which strengthened each other's claims, we think. So how am I doing for time? So patent triggered immunity is usually activated during ETI investigations, as I've said before. So if you transiently express with Agrobacterium or you deliver Pseudomonas, you're actually activating PTI plus ETI, because Agrobacterium is full of PABs, same as Pseudomonas. But estradiol inducible system means we can activate ETI in the absence of PTI. So what happened, and this is an old idea, but before it was used more for genetics than for physiology. So one thing we were able to show was that if we pre-treat with estradiol and then provide Pseudomonas syringae strain DC3000, which causes plenty of disease on Arabidopsis, then the amount of bacterial growth is reduced. We call this the super ETI line for the inducible ETI. That's what SETI means. So if we've introduced with estradiol, then we get greatly reduced bacterial proliferation compared to if we haven't induced with estradiol, or compared to just putting estradiol on Columbia. So the system works, and it works in that way, in that sense, as we'd expected it. If we pre-treat with estradiol and then activate PTI using classic ligands like flagellin 22, which is recognized by the FLS2 immune receptor, we can see that with ETI plus PTI, estradiol then flag 22, we get a bigger burst of reactive oxygen species, and it also persists longer. And you see the same sort of thing quantitatively depicted here. So in that sense, you can see already that ETI potentiates the strength of PTI. Here's another illustration of that. So if we infiltrate Pseudomonas, or actually in this case, this is a mock, we don't infiltrate with anything, and do diaminobenzidine staining, which detects hydrogen peroxide, we don't see anything. So and starting with the control here, if we infiltrate Pseudomonas mutant that can no longer deliver any effectors, this is type 3 secretion mutant, after 2 days, then there's no DAB staining. If we'd looked after 1 hour, we would see DAB staining, but it's all gone by 2 days. ETI alone, no staining. But if we have estradiol inducible plus the Pseudomonas, after 2 days, we can see there's quite a lot of DAB staining. And I think probably this next set is also quite illustrative. So if we infiltrate this heart mutant of Pseudomonas, which is deficient in type 3 secretion, doesn't grow at all, infiltrate leaves of Arabidopsis, and we look with protein gels at the amounts of BIC1, a receptor like cytoplasmic kinase involved in immunity, or the RBOHD, which is the respiratory burst oxidase that makes reactive oxygen species, or MAP kinases, or we look for phosphorylated MAP kinases with this antibody. This is what you see. But if you've infiltrated with a functional Pseudomonas that can cause disease, you can see there's attenuation of the amount of activation of this, and of the amount of activation of this, and of the amount of accumulation of RBOHD. So this is part of the action of the effector repertoire of the Pseudomonas. You reduce the hallmarks of the plant defense response. However, if you've pre-treated with estradiol and then give it the Pseudomonas, guess what? You get a big fat band for BIC1, a big fat band for RBOHD, a greater MAP kinase accumulation, more persistent MAP kinase phosphorylation. So again, this is and this is true for multiple effector triggered immunities, so AVR-RP2, AVR-RPM1, etc. It's in the paper, and also for multiple different PAPs. So conclusion then is that what ETI does is it reboots PTI, but actually it's PTI that's stopping the pathogen. So the prediction then might be that ETI won't work in a PTI mutant. So to do it another way, here's the zigzag-zig. Pattern recognition receptors detect some things, a lot of things. Effectors drag that down. But what NLRs do is to restore the PTI. So if you have a PTI deficient mutant that's depleted, knocked out in these two helper receptor-like kinases, BAC1 and BKK1, that support the action of FLS2 and EFR and others, then what you see that even if you've pre-treated with estradiol to induce AVR-RPS4, you get very little enhancement of resistance in terms of restricting bacterial growth compared to wild type. So in wild type, you've got this, you inactivate PTI, then estradiol no longer gives you that enhancement of resistance. And also, if you look at an FLS2 EFR double mutant, you get the same kind of result. You get basically the estradiol-inducible activation of PTI doesn't work because PTI is damaged. What about the other way around? So I mentioned before, so if you induce with estradiol and look for an HR, you know, from ETI alone, you don't get it. But if we combine estradiol, well, look at it this way. If there's no estradiol and we give it DC3000 Hard C or Pseudomonas fluorescens or various kind of PAMPs, the mix, again, we don't see an HR. But the combination of ETI plus PTI gives you that HR. And this is true for multiple PAMPs and multiple inducible effectors. So, we also showed that with various mutants and other tricks, that for this HR, the cell death to happen, we need functional MAP kinases and respiratory burst oxidases. So here's the bottom line. We got this, we sometimes say the zigzag-zig turns full circle. So pathogens come in, you get PTI, effectors suppress it, but if an NLR is activated, the NLRs restore it. So pathogens activate PTI and it can shut them down unless their effectors suppress it. But if those effectors are recognized by an NLR, ETI restores the PTI. So ETI, we know that ETI elevates the gene expression and the protein abundance for these. It's not all gene expression. I think there's a translational component mechanistically. But PTI is the primary source of resistance, and ETI restores PTI after effector attenuation. And ETI is receptor limited. Back to make this point again, our genes are semi-dominant. So here's, I think, a crucial point. So multiple NLR recognitions will have not just a genetic benefit in that the pathogen has to lose multiple effectors, recognized effectors to overcome it, but also it has a physiological benefit because you've got more recognitions, then you just completely stop the pathogen dead. And if you've got a weak resistance that the pathogen can grow on a bit, you can imagine it's easier for the pathogen to evolve a variant that can evade that resistance. But if the pathogen is absolutely stopped dead, it's squelched because there's so much resistance, so much defense mechanisms activated, then the pathogen never gets off the ground again. The pathogen it's put on the mat and it stays there. And that, in a sense, is what non-host, that's my interpretation of non-host resistance. So I'd like to thank my colleagues on the Phytophthora infestans resistance side, Camille, who started the RENSEQ with Florian, and Zhao, and Robert, who've really advanced it, and Agnes, who's been doing our field trials and varietal improvement with transgenic potato lines, Andrea provided crucial support to Zhao. And then for the, and Sebastian was also part of that project. And then Bruno and Ping Tao and He Kyung played a crucial role in that mutual potentiation story that I've just told you about. I'd like to thank all the diverse funders, the Gatsby Foundation that funds the Sainsbury Lab, European Research Council, BBSRC. I'd like to thank various colleagues over the years who've gone on to start their own labs or long been in their own labs, colleagues at TSL, colleagues in the support teams, and you for your attention.
So, thank you, Professor Jones, for a very enlightening talk, and especially your idea of making our future crop non-host for the commonly invading pathogens. So I'm sure many of our audience, many from us are having queries which they listen to more, or they trying to interact with you. So I have requested them to place their questions in the chat box, and I find some of them here. So first from Dr. Mandal, and he tells that more than 140 NLRs with known recognized effectors have been identified. Cross-species transfer of NLR stacks provide durable resistance against pathogens. And how do NLRs activate downstream signaling components?
Yeah, well, that is an area of very active research in many laboratories. I think we've gone as far as we can with genetics in terms of forward genetics. So we know that, for example, I referred to helper NLRs. So there are many helper NLRs, or sorry, I distinguish between the sensor NLRs, which detect the pathogen, and the helper NLRs that are required for signaling downstream. Now, there are some sensor NLRs which appear not to need any helper NLRs. So RPI-VNT1 is in that category. And it has homology at its N-terminus to the ZAR1 protein that makes cation channels upon oligomerization. So I think that that's probably what RPI-VNT1 does. Now, there in Arabidopsis, at least, there are the ADR1, NRG1 helper NLR class. And if you have a pentuple mutant, if you knock them all out, then again, there's quite a few, all the TR NLRs and several of the coil-coil NLRs do not work, but RPM1 still works. So they differ in their requirement for helper NLRs. I think basically if the sensor NLR is incapable of making a cation channel, then something happens which means that the helper NLR does it for them. Why is calcium influx, it's almost certainly calcium influx, sufficient to activate defense or to restore attenuated PTI? Why, where in the cell does it happen? These are areas of active research. We have a couple of papers we're submitting soon on that topic about how the helper NLRs work. The NRC helper NLR and the NRG1 class of helper NLR seem to also get oligomerized upon activation. But what happens after that, I don't know. So I talk about transcription induction being part of it. So it could be that, well, there, in Arabidopsis at least, there are quite a lot of transcription factors that carry calcium regulated domains. So there's the calmodulin regulated transcription factors, the CAMTAs, and there's also CBP60, which have EF hands that bind calcium. So that might be part of it, but there's also a translational component, and we've got no idea how that works. Maybe there are some things that regulate translation that are sensitive to calcium levels, but I haven't seen those reported. So short answer, good question. Such a good question. A lot of people are working on it.
Yeah, thanks, Professor Jones. We have another question that how to crack the structure of the domain of RRS1 or RPS4 protein to resolve the mechanism completely?
Yeah, well, again, that's a matter that we're rather interested in. So we did recently report in PNAS the structure from a very talented Indian postdoc, Natika Mukhi, who's now gone to the Max Planck Institute. So that she had a first author paper in PNAS last December on the co-crystal structure of AVR RPS4 with the Werke domain of RRS1. And so how does that binding then get translated into activating the whole paired immune receptor? We have a collaboration with Gigi Chai and his team who are trying to express RPS4 and RS1 in insect cells with the goal of getting cryo-EM structure on that paired NLR. But it's, it's one of these things where it's a bit of a dark art to successfully make protein that can be subjected to a structural analysis, and that's a work in progress.
Okay, so Dr. B.L. Patil, we have a query from Dr. Patil. So he asks that how fast the plant R genes evolve to counter the pathogen virulence genes?
Yeah, it's I think you often see in the literature that they're rapidly evolving, and I think that's a bit misleading because there's no real evidence that they have a mutation rate that is higher than any other gene. Although I think we want to look again at that in the light of Detlef Eigel's very recent paper in Nature in January about how mutation rates are different across different parts of the genome. But I think what I come back to this thing, remember that so-called SMARTY model with all the different colors to represent the genetic diversity and recognition capacity in natural populations. So a lot of that diversity, it's standing variation, but the alleles actually diverged a long time ago, but they're maintained because they work, provided they're rare. But basically most R genes work really well when they're rare, but when you have a monoculture, then you've got much stronger selection for the pathogen to overcome them. So the short answer is I don't think NLRs evolve at a particularly high rate, but there's been selection over millennia for diversity in NLR repertoires in natural populations. And so when we see a lot of differences in alleles, NLR loci, that's why. It's not because they're evolving rapidly, it's because they've been selected for diversity over a very long time.
Okay, so the next one is, what do you think, why plants do not have an adaptive immunity?
Well, adaptive immunity is something that was invented, I was going to say once, it might be 2 or 3 times, in animals. And what the animals did was to steal the activity of a transposon. But key to the effectiveness of adaptive immunity is the fact that there are mobile defender cells. And there are defender cells, the B cells, if you've got a B cell that can recognize, let's say, COVID, and then you get COVID, you get a massive expansion in the population of those B cells that respond to COVID, and then they travel throughout the body in the bloodstream. Now, plants don't work like that. Every little cell is in its little cardboard box, and it ain't going anywhere. So, it's just sitting there, and it has to be able to cope with whatever pathogens life throws at it. So, yeah, I think that's why there's no adaptive immunity in plants.
Okay, so I can see some more questions. So if your time permits, so shall we take some of those?
Sure. Yeah, I've got. I don't have there's nothing else I need to. I've got another 10 minutes at least.
Yeah. Okay. So the next one is there a clear boundary between PTI and ETI? And does the zigzag model of R-AVR interaction takes this aspect into consideration?
Well, I mean, I tried to address that in my talk. So I think all I could do is restate what I said, which is that, I mean, the main difference between PTI and ETI is that one is initiated by cell surface receptors upon detection of outside molecules, and one is initiated upon detection of inside molecules or the effects of inside molecules. But the actual immune response is very similar. I mean, the difference between PTI and ETI, at least for the TRNLRs, is that the TRNLRs activate salicylic acid biosynthesis to quite a high level. And of course, it's a bit more complicated. Yeah, life always is than the simple story you want to tell in a simple way to get your main point across. So virus resistance, are there any PAMPs in a virus? The N gene of tobacco confers resistance to tobacco mosaic virus. And what you do know is you get a lot of salicylic acid made and moved around the plant. So it's a hormone that's mobile. During the activation, the infection of a tobacco plant that has the N gene that confers resistance to tobacco mosaic virus. And you see these local lesions that restrict the pathogen. Now, is there any PTI there at all? We don't really know. I think that's quite an interesting thing to investigate properly for somebody. But, I think it's worth revisiting in the light of our enhanced understanding of the relationship between ETI and PTI. Yeah, and in fact, you can also note, Nirvana, that there's quite a few recent reviews from my co-authors, Bruno and Ping Tao. This one just came out in Plant Cell on some topic. So yeah, you might find those useful because we go into this in more detail.
Okay, so the next one is how is the advancement in understanding of plant susceptibility genes vis-a-vis the R genes, as they can be easily made resistant by genome editing using CRISPR-Cas system of susceptibility genes?
Yeah, no, that's a good question. So of course, let me say something about what I didn't say anything about. I didn't talk about S genes. Also, I didn't talk about pathogens like botrytis, the necrotrophs, where it's less clear that there are effectors that are recognized. There's no dominant resistance genes to botrytis. There's not much genetic variation, and where there is variation, it's mostly attributed to antimicrobial compound biosynthesis variation. So I think for some pathogens on some plants, S genes are a very important path to resistance. And one of the most beautiful examples, of course, comes from rice. Maybe some of the co-authors are on the line, where they CRISPR'd out 3 different TAL effector targets, or at least the promoters of 3 different TAL effector targets that encode sweet proteins, such that the resulting triple mutant CRISPR plant of rice was more resistant or fully resistant, because the TAL effectors did not work to enhance the expression of sweet proteins, which contribute to bacterial success. So I think I did not intend at all to indicate that that there are no other ways of addressing these issues. And indeed, if you combine S genes with R genes stacks, then you'd be even more confident in the durability of the resistance you come up with. So I think, I like to quote Sophie and Kamoon on this one. So, somebody asked Sophie, and how can you make sure you get durable resistance? And he said, well, we can't make sure of it, but what we can do is we can make sure we avoid paths to susceptibility. We know we're very confident in what will not give us durable resistance. So if you've got 3 different resistance genes, for example, that recognize the same effector, then it's going to be pretty easy for the pathogen to mutate that effector, and then it overcomes 3 different resistance genes. So, and you do see examples of different resistance genes recognizing the same effector. So back to your question, S genes are also important.
Okay, so next we have Amy Redker, who emphasized, who tells that you emphasized the RPI AMR1 and AMR3 recognizing P. parmivora effector as well. Do you plan to test the stack of these 2 RPI against any of the root pathogens?
Yeah, good question. Thank you, Amy. So what we know so far is that RPI-AMR3 confers good resistance to Palmivora and Parasitica, but not to every race, actually. I think it's like 5 out of 7 races that we tested, Palmivora, maybe the same with Parasitica. The most exciting thing the thing I'm most excited about testing next. So we know that, for example, the BS2 gene that I showed earlier out of pepper, you put it into tomato, it works. It also recognizes a Xanthomonas effector for Brassica. Put that BS2 into Brassica, it doesn't work. So we've now got a good hypothesis about why that might be. Well, let me take a step back. So strawberries, for example, the strawberry pathogen Phytophthora cactorum carries AVR-AMR3. So basically, I think what we need to do is to move the sensor NLR and the helper NLR. So what I'm most excited about doing is testing whether, if we take, let's say, RPI-AMR3 and one of the NRCs and put that into strawberry, we will now get resistance to Phytophthora cactorum. Because I think for things to move NLRs from one plant family to another, you may also need to move the cognate helper NLR.
Okay, so one more question from Devana. So he asked that what is your take on wheat blast and rice probably as a source of non-host resistance?
I'm not an expert on this, and my colleagues Nick Talbot and Sophie Kamoon are experts on this. I think it's, if you can define effectors that are recognized from wheat blasts that are recognized in rice, and you can chase down the corresponding gene, then I think it's a very good idea to test in wheat whether it works. And the same is true, for any recognition in barley or any other grass, actually. And I think my colleagues are actively investigating this possibility, but you'd have to ask Nick and Sophie where they got to.
Okay, so another one from Dr. Vishwanathan, that our genes are not clearly defined in many cultivated crops. Similarly, the PAMPs of their pathogens are also not known.
Well, so what you see depends very often on how hard you look, and also what phenotype you're monitoring for. So, I mean, there's a lot of R genes in rice against rice blast and against Xanthomonas, as I understand it. So I think there's a lot to work with in rice. I think rice is a very good system, actually. Doesn't grow very well in Norwich, though. But of course, there are some plants that have not been subjected to such intense scrutiny with genetics. So, there may be R genes there that nobody's seen yet. PAMPs, well, there are, pretty much every plant responds to flagellin and to chitin. And there may also be more pathogen-specific recognized molecules that are recognized by surface receptors. But again, you actually have to do the investigation. So some of my colleagues, for example, are looking for PAMPs from the soybean rust pathogen and looking for receptors that might recognize them. So, you know, it's seek and ye shall find.
Okay, so probably we are having the last one here in the chat box. So from Dr. Ruchi, and she asks that where do the recessive R genes stand in this whole scenario? And with a very little background on these the aspects of host plant resistance, I also want to add a note from my side with that you have already mentioned that these R genes are semi-dominant in nature, and stacking more and more R genes is likely to give better resistance in the plant. So what is your take on that? So do they have any other physiological implications, stacking more and more number of genes, and what is the maybe ideal limit for that?
Well, can I come back to that? I think I'll address Dr. Patel's question and then come back to your one there. So recessive resistance genes, I know that, let's say XA13, is that a rice recessive resistance gene? That turns out, I think, as I recall, to be a sweet protein. And there's certainly some of the other ones are. And there's also, there's a recessive resistance gene to blast. Was it PI23? Anyway, that was a loss of function of one of these HMA domain proteins. So clearly they will be helpful and indeed should be included as far as possible in the breeder's repertoire. They're rarer. It's very important to understand the mechanism of them. It's quite interesting that the HMA domain protein, is it? Maybe it's PI19. Anyway, I don't remember. It was an HMA domain protein, and that same kind of domain is integrated into several NLR-ID pairs. So it tells you that something about host targets. So you can enhance resistance in two different ways, and they're not mutually exclusive. If you can do both, that's got to be better. So you can have a detection-based NLR-ID, such as with HMA domain in RGA45 or PIC1, PIC2, or you can have an absence of target strategy, so the pathogen is less effective at making the host susceptible. So from the standpoint of providing a solution to a pathogen problem, if you can use both, it's better than either alone. Dr. Mann, I've forgotten. If you have stacks of R genes, is that a problem? So think about it this way. So diploid, or inbred diploid potato has 750 NLR genes. A tetraploid potato, where at each locus there's usually 4 different alleles, therefore has 3,000 NLR genes. So if you add another 2 or 3, you're not going to have a huge impact on the physiology. So that's my general answer. And then, of course, if you use the GM method, you can get outcomes that you have to screen out. But you get plants that are a bit stunted or whatever. So if you make 100 transformants, you'll find 5 that look totally fine, that look indistinguishable from wild type, and could be a variety.
Okay, thank you, Professor Jones, for the wonderful talk, and also very patiently addressing all of the queries from our audience. So I'm sure that our audience have, our colleagues, they have tremendously benefited from your talk, as well as the opportunity to interact with you, which they have got today. So
My pleasure.
thank you. So before we close today's program, so I would really, it is my great pleasure to listen to Professor Jones, and with the now we are concluding our today's special talk, which was on dissecting and deploying the plant immune receptor mechanisms for crop protection, by extending hearty thanks from whole NRRI family as well as from ICAR to Professor Jonathan Jones for accepting our invitation and delivering the special talk. Sir, your talk was so fascinating, and it has surely ignited our minds to think on the aspect you just presented. Also, this is a remarkable opportunity for us to listen and interact with you. So once again, thank you very much. And I hope our stronger association with you, there would be the stronger association with you in future. We are grateful to have you today with us. So I take this opportunity to express our sincere gratitude to our respected director, Dr. Padmini Swai, for presiding over today's program. So unfortunately, she had to leave for one of our council's very important meeting. So, and for her constant support and guidance to make this program a success. Thank you very much, madam. I also thank Dr. P.C. Rath, the principal scientist and chairman of organizing committee of this program, Azadi Ka Amrit Mahotsav at NRRI, for planning and coordinating all the activities. I'm also thankful to Dr. Sudhamay Mandal, the principal scientist and the convener of this program, for meticulously planning and organizing the event. I'm thankful to all of the heads of divisions as well as our regional stations, scientists and technical staff, and all other staff and students of this institute for attending today's special talk and also providing necessary support. I express my gratitude to all the directors, scientists, researchers, students, and office staffs of other institutes who attended this program on this virtual platform. I thank the staff of our cells and also the committee members of this Azadi Ka Amrit Mahotsav committee at NRRI for making all the necessary arrangement for successfully organizing this special talk. Thank you, and with the permission of the chair, I am concluding today's program here. So Professor Jones, thanks once again, and also requesting you to respond to our emails for any clarification and or guidance in this field. Thanks, and stay safe. Thank you.
Thanks. My pleasure.
Thank you.
Have a great evening.